Hello everyone, I'm Ritika Sridharan and I will be presenting our research on interpretable cervical cell classification, a comparative analysis. This research was conducted by Nishantini Gnanavel, Pradushan Inparaj, Professor Dulani Medenia, Dr. Pradeepan Yogaraja and me. Though cervical cancer is the fourth most common cancer among women, it can be treated easily when detected early. Therefore, it is important to focus on precise screening methods to better help those at risk. Traditionally, the pap smear test and the liquid-based cytology test have been used to identify cancerous and precancerous cells. However, manual assessments are time-consuming and labor-intensive with the risk of missing positive cases. To address these challenges, there is a critical need to automate the screening process. Though such automated systems were proposed and they performed with high accuracy, there is skepticism among clinicians to deploy these systems due to their black box nature. This issue motivates us to use explainable artificial intelligence or XAI techniques in this critical healthcare setting. XAI makes the model decisions transparent and understandable and therefore promotes the acceptance of such automated systems. Explainable methods also help to identify and rectify errors in models and enhance their overall accuracy. The existing studies employ several techniques such as transfer learning of deep pre-trained models, ensemble architectures, deep feature fusion, inclusion of handcrafted features, and feature reduction with principal component analysis or canonical correlation analysis to classify cervical cell images into normal and abnormal classes with very high performance. Similarly, we have listed certain studies that have applied different XAI techniques in healthcare settings. None of these studies belong to the cervical cancer domain. Also, most of these studies have overlooked the evaluation of chosen explainable methods. These limitations lead us to see the novel contributions of our study. To the best of our knowledge, XAI techniques like gradient weighted class activation mapping or GradCAM, GradCAM++, layer-wise relevance propagation or LRP have not been applied on cervical cell classification systems in previous research. Therefore, applying these explainable methods on this domain is a novel contribution of our study. However, we don't just stop there. We also critically analyze these applied XAI techniques both qualitatively and quantitatively to measure the faithfulness, stability, and interpretability of the generated explanations. Next, we will look at our methodology. We will go through each step in this process overview diagram more elaborately in the upcoming slides. Our study uses the Herlev dataset, which has 7 classes, but we reclassified this dataset into the 2 classes, abnormal and normal, for the scope of our study. First, we resized all our images to a uniform size of 224 by 224. Due to the inherent noise and low contrast of the pap smear images, we used median filter to reduce the noise and histogram equalization and normalization to increase the contrast. This better helped to extract important information from the images. Next, to increase and balance the number of images in each class of the training set, we applied several data augmentations, including translation, scaling, rotation, distortions, noise additions, and so on. Then, to classify these images into normal and abnormal classes, we used VGGNet, ExceptionNet, and EfficientNet that were pre-trained on the ImageNet dataset. We froze the earlier layers to capture the generic features. Latter layers were modified and trained on our dataset with the shown hyperparameters to capture the dataset specific features. Now, for the most important step of our study, which is the application of XAI, we use 3 different techniques: GradCAM, GradCAM++, and LRP. GradCAM captures the hierarchical features in the network through a forward pass. Gradients are computed through backpropagation. GradCAM++ improves the accuracy by integrating the first and second order gradients. And finally, the heat maps are generated by multiplying the normalized weights with activations. Next, when applying LRP, the top-level dense layers are converted to equivalent convolutions. For example, in our VGG model, the weights and biases of the first dense layer with 25,088 neurons are converted to the weights and biases of a convolutional layer of 512 kernels, each of size 7 by 7. After converting the image to a torch tensor, it is propagated through the network and activations of each layer of our modified model are collected. When the final dense layer of 2 neurons are reached, the predicted evidence is retained for the abnormal class. This retained evidence is propagated backwards to the very first convolutional layer of the network using propagation rules. For example, the set of rules we use here are designed based on the suggestions provided by Montavan in his paper on LRP. Finally, these propagated relevance scores are pooled over all the feature maps to visualize salient regions as a two-dimensional map. To evaluate the performance of our system, we will use accuracy, precision, recall, and F1 score for classification, image entropy, and pixel flipping metric for XAI. Accordingly, our classification metrics show that VGGNet and EfficientNet have performed well in cervical cell classification. From both, we chose VGGNet because it has a sequential architecture without skip connections, which offers a great starting point for LRP. Now we will visually inspect and analyze the explanations provided by our XAI techniques. In this slide, we have illustrated 3 images that were predicted correctly with the highest confidence. GradCAM and GradCAM++ activation maps have highlighted the regions within the nucleus and cytoplasm, which are the main indicators of abnormality in cervical cells. GradCAM++ activation maps have more regions within the nucleus with higher activations, and the highlighted regions are sharper and well defined than in GradCAM. But in LRP, only the nucleus has been outlined clearly, while the cytoplasm has been ignored. Anyhow, all these 3 explanations confirm that the classifier's decisions are based on nuclei or cytoplasm correctly and no other spurious feature. Usually, abnormal cells only have very large nuclei, but these normal cell examples shown over here also have large nuclei, and therefore, our classification model has been deceived by the large nuclei and predicted these incorrectly as abnormal. This way, we can understand the model's decisions based on explainable methods because GradCAM, GradCAM++, and LRP all highlight or outline the large nuclei here based on the model's decisions. For quantitative evaluation of our explainable methods, we first use image entropy. This checks the level of disorder in the pixel values of an image. Higher entropy value means a more complex representation that has not only relevant but also some extra features in the explanation. Lower entropy value, as for LRP over here, is preferred because it means the explanation is less random and has only the very significant and relevant aspects. The next metric we use is the pixel flipping metric, which simulates a perturbation by flipping the individual pixels from the ones indicated as most relevant to the least relevant by the XAI methods. Its aim is to measure the decrease in the prediction confidence as the relevant pixels are flipped. This steep drop in the LRP curve compared to the other curves indicates that LRP has identified the region most important to the model's decision correctly, since the significant perturbations in that region result in drastic changes in the model's decision and prediction confidence. However, for smaller perturbations with a lower number of pixels being flipped, the prediction confidence has not changed considerably, and this indicates the robustness of LRP. In conclusion, we have applied 3 different XAI techniques on our VGGNet-based binary classification model for the very first time in cervical cancer domain. While all three techniques properly highlighted the relevant regions of nucleus and cytoplasm, quantitative comparisons favored LRP with its lower entropy and steeper confidence drop for perturbations. Thank you.